AP Biology students, welcome to your 6th Prezi for Evolution, Speciation. This is definitely one of my favorite presentations in this unit, if not one of my favorites throughout the year. Big questions: how do we define a species? And how can evolutionary changes in a population lead to the production of new species? It's always a good idea before you take notes to have some questions written down so that your notes can answer them. In addition to these big questions, you can use the pre-discussion questions that I gave to you earlier in the year in hard copy or on Moodle. All right, so don't let the title fool you, The Origin of Species, because Darwin never actually tackled how new species arose. Instead, he was looking at how populations were different from each other. And so the origin of species has very little to do with the origin of actual species or how those species developed. So how do we define a species? So there's the biological species concept, which states that a population whose members can interbreed and produce viable fertile offspring. You definitely want to write that definition down for a species because I'll emphasize that a lot in class. That's not the only species definition there is. Morphological species, that would be based on appearance. Ecological, paleological, those might be based on niche or based on fossils. Sometimes there are alternate definitions needed, and you can brainstorm about why we might need these other few types. So go ahead and do that now. Some species are not alive anymore, and so it can be helpful to look at fossils or to look at morphology. And if you're not able to determine if species are able to produce those viable fertile offspring, sometimes other definitions of species are necessary. So the big thing that you should know, and you should write this down in bold, is that speciation, meaning the creation of new species, equals reproductive isolation. And so this is really important here. So species are going to be created by a series of evolutionary processes that result in the reproductive isolation of a population. And so what sorts of processes can isolate a population? There are two types that we're going to talk about. There's allopatric speciation and sympatric speciation. The first is a little bit easier to understand. The second takes a little bit more time and focus. So allopatric speciation, first of all, allopatric means other country. And so this is when a population is isolated from other populations due to physical geographic barriers. So if given enough time and enough evolution, the populations that are separated from each other will become reproductively isolated. What sorts of barriers or events do you think could contribute to allopatric speciation events? And so you guessed it. If there's an original population, and then the initial step of speciation is some sort of physical barrier or geographic barrier, what can happen over time is that now we have two distinct populations that are not breeding with each other. And so eventually that might result in reproductive isolation and potentially a new species, or there's two species becoming two separate species. And so if you try to then recombine those areas or recombine those organisms or populations, if they're unable to produce those viable fertile offspring, they'd be no longer considered members of the same species. So the other type of speciation is sympatric speciation. Sympatric means same country. And so this is whenever populations remain in the same physical area, but become reproductively isolated from other populations due to other mechanisms. And so again, this idea of reproductive isolation. Yes, they're in the same physical area of each other, but they are reproductively isolated from one another. So what sorts of events do you think could contribute to sympatric speciation events? So here's my original population. In the case of allopatric speciation, to review, we have some physical barrier separating our population of fish, and eventually, if given enough time, you will have these populations be reproductively isolated from one another, and eventually a new species may be created. Whereas in sympatric speciation, while there's no physical barrier here, there's some mechanism keeping organisms in keeping some of these fish from mating with or the other fish. So in some way, these fish are reproductively isolated. These two blue fish are reproductively isolated from these yellow fish, even though there's no physical barrier. Okay, so there are a couple effects of isolations. So this image can be really helpful to understand. And let's break down the text first, then we'll go back to the image. So isolation of a population, it doesn't always result in speciation. So just because you have two populations that are separated, doesn't mean there's going to be a speciation event. There are several outcomes that can occur following this isolation. And then depending on the circumstances, hybrid populations can serve as genetic links between isolated populations. This is typical of ring species, like these gulls in the northern hemisphere. So all of these are members of the same species of gull. So if you look over here, we've got a population. These blue little pieces here are populations, and these arrows represent gene flow. So gene flow is the opposite of reproductive isolation because we've got genes flowing in and out from various populations. And then if you look over here, we've got it looks like there's some barrier to gene flow. So this could be a physical barrier, or it could be a non-physical barrier to gene flow, resulting in reproductive isolation. Or maybe you could have populations that continue to have gene flow and remain the same species. Or you might have an isolated population that diverges. The word diverge means to become different than. So you can see how this population is diverging as there's no gene flow taking place between these other populations of the same species. And then here what we have is we've got this kind of murky hybrid zone which can take place. And so there are a couple possible outcomes for a hybrid zone. So what we could have is we could have only the loss of this hybrid zone is going to lead to speciation. And so one thing that we could have, a possible outcome, is that they would stay split. So we would not have this hybrid zone, meaning like a creature from this population and a creature from this population are reproducing, and we would have these hybrids. So we could have reinforcement, which would be like they continue along these paths of being reproductively isolated, and you can see the hybrid zone is narrow to the point of not existing. Or what we could have is we could have a fusion. Maybe they were reproductively isolated, but then now they're no longer not reproductively isolated. So that would be this picture here, this fusion. Or maybe this hybrid zone just kind of carries on for a long period of time until those populations reach what we call stability. So now we're going to talk about species barriers. Various mechanisms, aka barriers, exist that prevent successful interspecies reproduction. So note that successful reproduction here, we're talking about production of fertile offspring. So two organisms have to be able to reproduce a viable, meaning living, fertile offspring, meaning the babies that they make have to also then be able to make other babies. So we can split these barriers into two categories, and they relate to when they occur in relation to fertilization. So we've got pre-zygotic. So if you remember from freshman biology, a zygote is what's formed when an egg and a sperm combine in sexual reproduction. And so these are barriers that are prior to actually that fertilization happening. And then there are post-zygotic barriers. These are barriers after fertilization. A general spectrum, of all of the different pre-zygotic and post-zygotic barriers. You can also see there are more pre-zygotic barriers than there are post-zygotic barriers. It is going to be important that you're able to draw on a couple examples of each of these so that you can communicate your ideas clearly about what would or would not lead to a speciation event. So let's start with species that live in different areas would undergo what we call habitat isolation. So why are they reproductively isolated? Well, individuals of different species aren't even able to even attempt making a zygote. This is a prezygotic barrier because one of these species lives in the water, whereas the other lives on land. So just by virtue of them having different habitats, they're not even able to try to reproduce with each other. So another example would be temporal isolation. So these are species that breed during different times of the day, or maybe different seasons or different years. So temporal isolation is a prezygotic barrier where it's not even possible for them to have a mating attempt because the skunk up here mates in the winter, whereas the skunk down here mates in the summer. So they're two distinct species of skunks. They're unable to reproduce, one of the reasons being because of temporal isolation. So another prezygotic barrier would be unique behavior patterns and rituals that isolate species. So things that identify and attract members of the same species, whether it's a really specific call or a really specific mating dance, prevent interspecies reproduction. So this here are blue-footed boobies. They have a special dance that they do. The males have to lift their feet in order to attract females. So another bird species would not be able to reproduce with a female blue-footed booby because they specifically are looking for or genetically disposed to pick up on this blue-footed signal of that they're the same species. And then of course we have some maybe some mating attempts that would go on, but we've got some things happening that prevent a zygote from forming. And so we've got reproductive anatomy from one species that simply does not fit with the anatomy from another species. So for example, these snails here, they've got whorls that go the opposite direction. So opposing whorls have opposing genitals. So they literally, their parts do not match up. Here we've got proteins on the surface of gametes. Those are sex cells. Don't allow fusion of the sperm and the egg. So in order for a zygote to be created, you actually need to have the sperm and the egg kind of fuse together. And sometimes there are proteins on the surface that do not allow that fusion to happen. And so this is the case here with these organisms that live in the sea. Okay, so now we see some organisms that can't even try to mate, right? And then we see some organisms that it might be possible for them to mate, but there are other prezygotic barriers that prevent a zygote from forming. Okay, but now we have postzygotic barriers. So let's think about this. Post means after, right? So zygotic means after there's actually an egg. So these organisms, maybe there are some species that are able to almost or reproduce, but they're not able to successfully reproduce because they have not created viable fertile offspring. So maybe the hybrid is really weak, right? So maybe you have two different species that mate together and the offspring they produce is really frail and they don't live for very long. And so this is called reduced hybrid viability. So viable means life. So they don't have very long life, and so they're not going to be able to reproduce. And then here we have a hybrid who's sterile. So this is the case if you breed a donkey and a horse, which are separate species. If you breed them together, you get a mule. Since mules are unable to reproduce, that must mean that the donkey and the horse are two separate species according to that biological species concept. And so we call this reduced hybrid fertility. That makes sense because the hybrid is the in-between and it's reduced fertility, meaning this organism here is sterile. And then finally we've got hybrid breakdown, and this is the offspring of hybrid is going to lose hybrid genetic combinations over succeeding generations. So over time, that breaks down. And so all of those are pre- and post-zygotic barriers to interspecies reproduction. So remember, in order to have reproduction, we're talking about successful reproduction. And so pre or post-zygotic barriers are making it possible for these separate species to actually be considered the same species. Okay, so there are some speciation studies that have been done. And so speciation has been observed in nature and in the laboratory in all major lineages of life. And so there are a couple examples of speciation studies. See if you can identify the species barrier or barriers that are contributing to the speciation. We've got two different species of antelope squirrel, one for each rim of the Grand Canyon. So take a second to pause the video and quiz yourself on what type of pre or post-zygotic barrier you think this would be. Next up, we have the formation of the Isthmus of Panama that's led to the creation of 15 pairs of sibling species in the snapping shrimp genus. 2 pairs here are shown. We also have of the toad population of Europe shows that there's only a narrow zone where hybrids between the yellow-bellied and fire-bellied toads can actually be found. So that's a hybrid zone. This is actually pretty cool. So this is the hybrid growler bear offspring of a polar bear and a grizzly bear. And here we have allopatric populations of mice on the island of Madeira, different chromosomal fusion mutations. Modern bread wheat is a hybrid species with genetic contributions from 3 wild wheatgrass species. And experiments with transgenic monkey flowers show different pollinator preference for species flower colors. This is pretty interesting. This is one of those sympatric, potentially, speciation events. Sympatric populations of 2 flycatcher species are much likely to interbreed than allopatric. And mate choice preference in populations of mosquito fish are correlated to body shape between female and male. Body shape is correlated to predation pressure. Fruit flies raised on specific growth media show a preference for mating with flies raised on the same media. Observations in the field and in the lab show that color perception is important in maintaining cichlid species barriers. Species of a new species in the wild, this hybridization has also been seen in the lab. Okay, so pause the video for a second so that you can go in your textbook or look at the videos to review those specific examples. So when speciation happens, is kind of up for a debate in terms of how it happens. So the rate of speciation is an ongoing event. Does it happen gradually or does it happen rapidly? And maybe both of those things is correct. So the thinking in gradualism, and this is kind of old thinking, which doesn't necessarily mean that it's wrong, is that Charles Darwin and Charles Lyell thought that gradual difference and divergence over long spans of time. And so big changes could occur as the accumulation of many small ones. So here we've got the original species here, and at the end of this time frame, we have 2 distinct and separate species. And after several small gradual changes, it's resulted in really big differences among those species. So that's gradualism. The next is punctuated equilibrium. And so this is what we've seen a little bit more in the fossil record. And so this is kind of newer thinking. And this is that the rate of speciation is actually not constant. Rapid bursts of change followed by long periods of little or no change. Species undergo rapid change when they first evolve from a parent population, often whenever there's little competition. So we've got punctuated pattern here. We have a pretty dramatic change, and then for a long period of time, there are no new speciation events within that lineage. All right, so if you look, this would be what gradualism trees kind of look like, whereas this is kind of what punctuated equilibrium looks like. So the idea here is that you go for longer periods of time without a speciation event. All right, everybody. You need to be able to make sure that you can do these things. So apply the biological species definition and identify circumstances where it is not applicable. Explain the circumstances that can lead to the production of a new species, both allopatrically and patrically. Define all species barriers described in this presentation and provide examples of these barriers. And finally, compare the common models of the pace of speciation and cite evidence that supports both gradualism and punctuated equilibrium.